I've gotten no confirmation that Aurora's fixed in this room. So if you want a recording, I suggest you just record the audio on your cell phone, because you're allowed to record me audio anyway, the audio. So I don't know why you're worrying about Aurora anyway. If you come to class, you can simply record me. As if you read the syllabus, it says that I allow you to record me audio record me for your own usage. So I would do that because Aurora's not showing up yet, and they control everything. So if they don't show up, apparently it was down in many rooms, and they weren't even sure they would have it fixed for when they would have it fixed. And they're also renowned for forgetting to record lectures. Okay, so this was epidemic in the fall, where instructors had to re-record when they had asynchronous lectures. I do not, so I do not and will not be re-recording the lecture, because I'm doing that for you, and you can sit in class. Okay, so as of right now, Aurora is not on. All right. Okay. Yeah. Okay. So a couple other things. So you've now gone through your first lab. You've seen how the lab is set up. Remember, the next 2 weeks will be a staining exercise to which you're given an unknown bacteria. And as you learn some new stains like gram stain, capsule stain, plus utilizing the hanging drop stain, the preparation that you did, and a couple of other very simple tests, you're going to use a flow chart to identify the unknown. So please make sure you fully read it. You can already construct your flow chart, look at the unknowns, and then separate them out based on gram negative, gram positive, and the tests that will lead to an identification. That will help you tremendously so that you're not under stress in week 2 when you forgot to do it and you got to do all of that at the end of week 2. In reality, you're handing in 2 slides, a gram stain and an endospore stain. The endospore stain you will do next week, and you will show your TA the capsule and the hanging drop. You have 2 weeks to do that. That is actually completely doable in 3 hours, but you have 6 hours basically to do that. So for the most part, most students do finish early in the second week because you're only really doing handing in 2 slides. But the key is to please make sure you've read the manual so you fully understand what you're doing and you can get started right away. All right. Okay, so today we're going to continue where we left off last week, which was talking about a variety of very important scientists who really did allow us to develop microbiological techniques. And we got to Pasteur, and I talked a bit about what Pasteur did, and I said he actually designed this very unique experiment. It's called the Swan neck flask, which basically finally demonstrated categorically that microorganisms were in the air. And it's really a very straightforward experiment, understanding that, this was more than a couple hundred years ago. Basically, he took a flask and he did some glass blowing and he connected a very long, thin neck. And it has bends in it, as you can see. And then he added the liquid media and he heated it, which is the equivalent of sterilizing it. And basically, he sat this flask out. And as long as this flask sits in the upright position, and the key is that air can move in and out because most bacteria, at least those that are anaerobic, require air, and the majority of them are anaerobic. What would happen is that the dust and other particulate matter would get stuck in the bends, and this never actually showed the evidence of any kind of bacterial growth. Then, if you simply, so this is it, the material sitting here, if you simply tipped it so that we reduced the number of bends so that it would be on its side, then you would end up with bacterial growth, establishing that in the air there were microbes. And so that is something you have to also understand why we are having you learn how to do aseptic technique. Because everywhere you are on a surface, in the air, sits bacteria and fungi, fungal spores, etc. So if we want to be able to grow pure cultures, we have to make sure we're using good aseptic technique that allows us to grow the organisms we want and not grow the organisms that we don't want. So that's the whole principle of using the Bunsen burner and the various tools and baking them red hot, allowing it to cool, because if you don't allow the loop to cool, what are you going to do to the bacteria? Kill them, right? So these are techniques that everyone uses in microbiology laboratories. Sometimes now you might use plastic, because unfortunately science has become very plastic, and so a lot of things are one-time use only. So the loop is actually in a sealed piece of paper that you buy from a company. You use it once and you throw it away. But the problem is, if you've ever sat in a especially cell biology lab now, the amount of plastic labs are generating is utterly ridiculous, quite frankly, which is very toxic to the environment. So if you can reduce your plastic in a laboratory, then you're reducing your footprint, because obviously to make those plastic tools requires energy, which of course increases your carbon footprint. Okay. So what did Pasteur achieve, really? He did develop the specific methods for removing all organisms, which is called sterilization. So when you sterilize, you remove all living organisms from whatever you're treating. He also developed pasteurization, hence his name, Pasteur, pasteurization. It removes only specific target organisms. So dairy products are pasteurized to remove some of the more problematic bacteria that are present on the dairy cow, but it does not remove all the bacteria. In fact, there are good bacteria in dairy products as well. Hence why once that here, almost all milk is refrigerated. If you buy milk in Europe and other locations, where might you find milk? in a farmer's market. Well, you can find that here too, but in terms of a grocery store, where might you find it? Yes, but there's another place you find it. Sorry? It's not refrigerated. It's actually in little boxes, at room temperature, because it's undergone a very specific type of pasteurization that allows it to sit at room temperature, but as soon as you open it, it has to go in the refrigerator. Okay. You can find these in Costco, flats of chocolate milk that are not in the refrigerator because they've been treated by this particular type of pasteurization. All right, so pasteurization is the removal of specific target organisms, but not all organisms. And of course, you've already experienced what aseptic technique is, trying to make sure you're only dealing with the organisms you want. And we also have to know what a pure culture is. So a pure culture is a culture that contains only one microorganism, which means you will have had to treat that media to make sure it's sterile, make sure you're using your aseptic technique, and then you actually put in the particular organism you're interested in. So this is obviously a method for isolating and studying all organisms, and very important if you want to study disease-causing organisms. And really, with the development of these techniques, along with a few others that became important, along with Robert Koch, who's another very important player in giving us tools, we really started the era of them looking for the types of microbes that causes disease. So remember, sterilization, removal of all living organisms. There's many ways you can do this. You can use a very small pore filter to remove microorganisms. You can expose it to heat, most commonly for microorganisms, for dealing in microbiology. That's going to be an autoclave. You can use ozone, you can use radiation, you can use a variety of halogens and other antimicrobial agents. Really, the method you use depends on the substance being sterilized. So for example, fruit and many vegetables that travel by truck from long distances are irradiated on their surface to try and reduce the number of microorganisms that are on the surface, because it takes them a while to get here from California. And if they didn't do that, they would already be bad. But everyone will have experienced buying what looks like a nice fresh set of raspberries or whatever, and then one day later they're all bad. Or my favorite thing is buying clementines at Christmas time. You buy a nice big bag of clementines, and within 3 days they're covered in mold, because that is dealing with surface organisms, but they have to actually do the entire surface to get rid of those organisms. All right, so the most common method of sterilization is what we call autoclaving. Really, this is really just a great big huge pressure cooker. So if you can imagine a pressure cooker on steroids, it's a very dangerous machine. What it does is it uses hot saturated steam under pressure, 121 degrees Celsius, with a pressure of 15 pounds per square inch. So you really don't want to come in contact with what's inside an autoclave if you don't want to go through what would be a very hot massage, which would result in you losing all your skin. Okay. All right, so under these conditions, most cells will be killed. But really, this temperature and pressure is really a ballpark, because really you have to make sure you actually destroy the spores, like the endospores that bacteria form, that you'll learn about in lab and we'll talk about later in term, because they are actually highly resistant to temperature, and also the spores of fungal cells. So while this works for most simple small volumes, the length of time needed for sterilization really does depend on the volume that's in the autoclave. So for example, if you have 2-liter amounts, like in 2-liter flasks, you can use 15 minutes. If you have a great big carboy that has 10 liters of liquid in it, it has to be autoclaved for 3 to 5 hours to make sure you've removed the organism. So this is the one thing that often happens in research labs. People don't know that you often have to scale up if you're actually autoclaving large batches of liquid in one vessel. All right, so all the media that you receive, that you inoculate, so next week you'll get fresh media to inoculate your unknown. All of that has been sterilized. The bacteria that are growing in tubes or on tubes, the media was sterilized and then inoculated by the technician. So again, in pasteurization, you're removing unwanted target organisms that can cause disease or rapid spoilage. And again, the conditions depend on the target organism. But the other important point is you're actually treating foodstuffs. So you don't want to destroy, if you were to put milk in an autoclave, you would destroy all the proteins, you'd end up with curdled milk, and it wouldn't be any fun to drink or do anything with. So you have to actually make sure you're not damaging the substance that is actually being treated. And really, Pasteur first did this kind of pasteurization on wine, so the French were dealing with spoilage of wine, and what he found was a very brief heating from 55 to 60 degrees would get rid of the problematic organism that was causing the wine spoilage, but would not affect the taste of the wine. If you went higher, then in fact you started to destroy the taste of the wine. So this is, or for longer time. So this is an important concept that really when you're doing pasteurization, you're trying to keep the foodstuff edible or drinkable while removing the bacteria or other microorganisms that would be problematic. So wine was actually, interestingly enough, the first foodstuff to be pasteurized. Milk and beer followed, first in Germany and then in North America. And the problem with raw milk, and a lot of people talk about drinking raw milk now and say it's totally safe. Raw milk is not safe. It has a lot of microorganisms that can make many people, including small children, the elderly, and the immunocompromised, sick because it has bacteria like Listeria in it. And as a result, it is really not a good thing to do. Listeria will be destroyed by pasteurization, but the beneficial organisms like lactobacillus, which of course is really one of the very positive bacteria. Lactobacillus is found in dairy products, very good for creating a good gut microbiome. In high numbers in yogurt, are not destroyed. So you want the good bacteria to survive, but you want the disease-causing bacteria to be removed. And really, most likely, if you were on a farm and you collected the milk and you drank it right away, it wouldn't be problematic because obviously there was no time for the small number of bacteria that would have been collected to cause real damage to you. But if you're going to a farmer's market and they're selling you raw milk and you don't know for how long it's been there and it's 2 to 3 or 4 days old, it's not particularly a good idea. So there's 3 types of temperature pasteurization, which is very interesting. You can see the temperatures go up from 63 to 150 degrees, and the timing actually changes. And really, the milk products that can sit at room temperature go through this ultra high temperature treatment of 150 degrees Celsius for 1 to 3 seconds. And in reality, if you taste dairy products that are fresh in the fridge versus this, there is a difference in the taste. Okay, so there has been some alterations in the milk. But what most companies now use, and you'll see it on a lot of the containers, is the filtration. So you'll see that a lot of the major milk products you buy in the refrigerated section right now, the major milk companies that you will find here, almost all of them are filter pasteurized. So they're using filters to remove the bad bacteria, but leave the good bacteria alone. And the reason the filtration is more positive is it really does not alter the component of the food stuff. Okay. You can also use exposure to chlorine. So chlorine is used in various places to try and sterilize material. But the reality is there's not a lot of things that you can use chlorine for. Obviously, anything that's going to be ingested, you can't use chlorine for. Chlorine is mostly used for swimming pools and a variation, and in fact, this still doesn't, in a swimming pool, the amount of chlorine is there, does not sterilize the water. They just have to keep the number of bacteria below a certain number, okay? The UV radiation at 260 nanometers. So what is the importance of 260 nanometers? What does 260 nanometers destroy? Anybody? DNA, yes. So this will actually destroy the DNA of the organism. And so this is a very good way to treat spaces in your tissue culture rooms. So biosafety hoods or other types of hoods all have UV lights on them. You turn them on, you irradiate that area, and it will kill the bacteria that are still on the, and it's stainless steel, kill on the stainless steel. It can't penetrate glass or plastic in short exposures, so it's not going to get into containers, but it will treat surface areas. There's also ionizing radiation, which is deep penetration that can sterilize plasticware, drugs, cosmetics, and some foods. So a lot of the plasticware that you buy and you work with in a laboratory is coming like Petri dishes that are in a sleeve. They have already been treated with gamma sterilization, so they're sterile. So therefore, all the bacteria have been killed. But the minute you open the Petri dish to pour media in it, it's no longer sterile. Okay, all right. And of course, your cosmetics go through this treatment as well. And this is one of the reasons why having microbiology skills is important, because quite frankly, cosmetic companies, food companies, food safety companies, as I said last week, Canada Blood Services, all need people who can actually test material for the growth of microorganisms to see if, in fact, they're safe for the consumers. So you've already experienced the aseptic technique minus the gloves, because there's no organisms you're working with that are dangerous to you. They're all relatively innocuous unless you want to sit and drink the whole tube of bacteria, and simple washing hands removes any contamination. And the reason this is not a great idea is because if you're not careful, if you get your glove a little bit too close to the Bunsen burner, it ignites. And then you have a bigger problem than some bacteria on your hands. So one thing that some of you, and I corrected some of you because it's important, but I'm sure there were a lot of other of you, your tendency is to stick the loop straight into the flame and sort of do this. You do it straight up and down as this person is holding it. So you want to make the whole metal piece of where the loop is red hot. Because when you're going in and out of culture tubes, if you only have done the loop, then the rest of that implement has not been sterilized, and you're going to transfer bacteria that you do not want to transfer. So it only takes about 3 to 4 seconds. It gets red hot. You allow it to cool. And then, of course, you can then streak for isolated colonies, as this person is doing with their loop. This is a key technique in microbiology, in medicine, in industry, in molecular biology when you're dealing with bacteria. Everyone who works with these has to use proper aseptic technique to try and prevent growing of microorganisms that you do not want to grow. Now, so if I say you have a culture in your hand, it means you have bacteria growing in a tube. If I say, and it may be the desired types, but it could also be multiple types of organisms. If I say pure culture, or you read pure culture, that means there's a single organism, a population of a single organism growing in that, on the Petri dish or in the tube. So in order to have a pure culture, first you have to remove all the organisms from the culture media, hence they get autoclaved for you, so they're sterilized. Then you have to inoculate, which is really an introduction of the organism, but the lingo of a microbiologist is to inoculate with a desired organism. Then you're going to incubate, which means provide them with the conditions they need to be able to grow. So that's going to be temperature and time and oxygen or no oxygen. And within that media, you have to give them their needed nutrients. So almost all the bacteria that you're going to use are very easy to grow. They grow on, as your manual tells you, either nutrient brought into nutrient media or Loria broth media, made up of peptone, some yeast extract, very simple media, has the building blocks for any bacteria. But some bacteria are fastidious, so they need very specific requirements for growth. Okay? So not all bacteria will grow in all media. So you need to know that. You need to know that some bacteria will have very specific growth requirements when you're working in a laboratory. And then once you grow them, you're going to see an increase in cell density that represents population size. So if you paid attention in the way you were given your samples this last week, were they on solid media or liquid media? Or both? Both. So you had broth culture, and then you also had a slant. So slants are the most ideal way to keep subculturing going, and you might say, well, how do I get the slant? Well, you pour the molten sterilized media into the tube, and you simply put it on a device that allows it to lie almost a little bit above horizontal, and it solidifies with the slant, which gives you a large surface area in which to actually streak out large amounts of cells. But you notice that neither of those techniques would allow you to get a single cell. You have a whole population of cells growing, and you're trusting that the person who did the work for you actually used the right techniques so that there's only one organism type. So when you're doing your unknown, if there's more than one bacteria there, you're not going to be able to identify your unknown. So the technician has to be using the right aseptic technique to make sure you get a pure culture of an unknown. All right. So what is the germ theory of infectious disease, which really still holds today? You know, if you read the literature, Robert Koch and Pasteur both did this independently, although Koch tends to get specifically credited with this in the late 1800s. And really, it comes down to a very simple statement. A specific microbe produces a specific disease in a specific host. And that may sound very easy to do, but actually it's not. So for example, Bacillus anthracis, so Bacillus is a gram-positive row. Anthracis, what do you think this species causes? It's named for the disease it causes. Anthrax, okay, produces the disease anthrax. And one of the things the reason I'm going through a variety of organisms' names is to make a point about the naming of most organisms. Oftentimes, their genus has some relevance to their shape, their species has some relevance to color, or the disease they cause. So really, the names are not that complicated if you understand what the names are telling you. So, for example, Neisseria gonorrhoeae produces the disease gonorrhea. This is Neisseria gonorrhoeae. It is a gram-negative cocci, but it's really a diplococci, and you can see it has the classic diplococci appearance of many diplococci where the two cocci come together, they have a flat side. Okay, so it's not two round cocci, there's a flat side. So this would have been produced as those two cells divided. They actually didn't. They created new cell wall, but they really didn't fully separate when they actually did their cell division. So once they had this idea of a specific organism being responsible for a specific infectious disease, investigators continued to develop the techniques that were going to be needed for doing this work. So just as another shape, so this is Vibrio cholera in a stain, but this is Vibrio cholera in a scanning microscope, and you can see that actually Vibrio is a comma-shaped rod. So it has this comma shape, and of course, it is the cause of cholera. Okay. So the names do make sense. I know you think you look at them and go, they all don't make sense to me. The majority of the names make perfect sense, and all the names you're going to see have some relevance to what they look like. All right, so the problem was they still needed a few techniques that we now use today. They needed a criteria for actually identifying these organisms because it wasn't really straightforward. So first they needed to have reasonable assurance that the specific organism was the causative agent. So they had to design a series of rules that are still used today in epidemiology. So first of all, yes, they needed sterile materials, they needed the right microbiological media, they needed growth chambers, incubators that could be set at different temperatures. so that you could grow the bacteria at different temperatures. They needed, when they started working with anaerobic bacteria, a way to remove oxygen from media and to remove oxygen from the incubators. They needed covered dishes, the classic plastic Petri dish. So someone had to develop that dish. Anyone want to guess or read who did it and why we call it a Petri dish? It was actually Robert Petri, okay? So they originally were glass, but now they're plastic. They're the only way you can identify a single colony, the only way you can. And of course, improved aseptic techniques and improved microscopes. So Van Leeuwenhoek would not have been able to see the bacteria very well that you saw in the lab. He would have seen something like what you saw under the 10x objective. But in order to really see the bacteria, you have to move to the 40 to 100, and you really need to be at the 100 oil immersion lens to really see the real shape of the bacteria. And of course, they needed staining techniques that would help them identify specific types of bacteria, i.e. the Gram stain, which differentiates bacteria into the two major groups. Based on the structure of their cell wall, they're either Gram positive or Gram negative. As a subpopulation of staining, they needed the acid-fast stain, which actually differentiates bacteria that have a very unique cell wall, that has a lot of lipid, which we'll talk about in much more detail in a later lecture and is in your lab, called mycolic acid. But the reason it's called acid-fast is the decolorizer is not just alcohol, which is used in a Gram stain to remove the stain from one set of organisms. It has acid added, which makes it a very strong decolorizer, which means only the bacteria that have mycolic acid will remain stained. And these are all taken for granted now, but people had to develop these stains in order to be able to do this work. So basically, the criteria, which is called Koch's postulates, was originally written for diseases of animals, but is still the standard for epidemiology. The caveat to that is that there are some organisms that we cannot grow. So we can't actually use these standard sets of rules. We would be using PCR to identify that the organism is there, because we can't grow them because they're too fastidious, or they might require being in the host. So there's some variations you might have to use with certain organisms. So the rules are really simple. You have a causative organism. It should only be found in the animals suffering from the disease, and should not be found in healthy individuals. The organism must be isolated and cultured, so you take that organism out of the diseased animal, you culture it, you characterize it based on staining, its shape, other biochemical techniques, and then you take that bacteria that is pure and you put it into another susceptible animal, so put it in another animal, and it should make that animal sick. And if it does, okay, then you should be able to re-isolate that organism from that second animal and see the same organism. And those are the rules by which diseases are actually, these are the steps that people would take constantly to try and make sure they have the right organism as the causative agent. That's just diagrammed here. Don't want to go in. I walked you through it on the previous slide, but really straightforward. You have the mouse, the healthy individual, you won't see the bacteria. Then you have plated a blood sample. You take off on the plate. You devour the red blood cells, which you're not going to see on the plate anyway. And you take the suspected pathogen. It should not be there in the healthy animal. You'll get isolated colonies. You pick an isolated colony. You put it back in the diseased animal. And then you remove a culture sample or tissue, blood or tissue sample. And again, you can show that the same organisms are present. What's not there is you don't just have the plate, right? You will take the isolated colony. You'll do a stain. You'll identify what the shape is, what the characteristic is. You'll do some biochemical tests on it to establish what its characteristics are. And then you'll repeat it when it's isolated from the second animal. Once that was established, they started to be able to identify bacteria that cause various diseases. And this is just a list to make a point also about names. And no, you don't need to know the year that they were discovered. But you do have to, this is a very small number of names, and you should know should be able to look at the name, and if I was to ask you what it is a causative agent, you should be able to tell me, because I've obviously given you very easy ones to do. So Bacillus anthracis, the causative agent of anthrax, Neisseria gonorrhea, Salmonella typhi, which is the causative agent of typhoid fever, Mycobacterium tuberculosis is the causative agent of tuberculosis, Mycobacterium leprae is the causative agent of, sorry? Leprosy, yes. Vibrio cholerae, causative agent of cholera, Clostridium tetani, which is a gram-positive rod, which is an anaerobic rod. It's why it causes such problems for us, is the causative agent of tetanus. And this is why you need what? Tetanus shot and a booster every, somebody said it, about 10 years. If you haven't kept up with it, if you show up in the hospital with a deep wound especially, they're going to give you a tetanus shot, because it's the deep wounds are the most problematic. Why? Because the deeper the wound, the spores can germinate in the deep wound, and in a deep wound, the base of the wound is anaerobic. Clostridium tetani cannot grow in aerobic conditions, but it can grow in anaerobic conditions, which means no oxygen. Clostridium perfringens was actually the causative agent of gas gangrene, which, if you know your history, was a major problem during World War I of infections in soldiers, actually. Yersinia pestis, which is another gram-negative rod, pestis, cause of the plague. So it was not the rats that caused the plague. The rats get a very bad name. They just happen to be carrying them around. It was actually the Yersinia pestis that actually caused the plague. Okay. And you can continue on this. So this is just kind of, and I give you some links if this is something that you're interested in. You can go to the Canadian Public Health Services. You can go to PHAC, which gives you information about pathogens. You can go to CDC or the World Health Organization. And really, we can start to see some of the diseases in the last, let's say, 35 years that we've started to identify. And some of these are what we call, and I'll show you a map in a moment, we call them re-emerging pathogens. So in other words, they've been seen before, they've disappeared, they've re-emerged, and they've generally re-emerged as being a much more serious infection. Like Mycobacterium tuberculosis, in the developed world, it was eradicated. It wasn't eradicated everywhere, but where it has reemerged as a serious infection as being a multi-drug resistant disease, which means it's exceedingly, it used to be easy to clear it up, the infection. Now it isn't, because it's almost resistant to all the antibiotics that are out there. And this is really a product, by the way, of people not taking antibiotics correctly. So, little lesson on antibiotics. When you take antibiotics and you need it for a particular infection, what usually happens within a day or two of taking the antibiotics? No, it still works, because you usually have to take antibiotics for 7 to 10 days, depending on the prescription you're given. But what do you feel like? You feel better. And so if you're somebody who doesn't have a lot of money, and you have an antibiotic you had to buy, and you start to feel better, what do? You say, I'll keep this for the next time I don't feel better. And that, of course, allows the bacteria to grow, to mutate, and become antibiotic resistant. That is why the prescription says take for usually 7 to 10 days. And if you still don't feel right, you need to go back to get another prescription, because otherwise you're setting yourself up for issues with that particular microorganism. All right. So we can take COVID-19. So COVID-19 what was classified, which is severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, is the virus strain that actually caused the coronavirus disease in 2019, COVID-19. But we had previous versions of coronaviruses. And one of the hot spots of a coronavirus, I skipped the MERS for a minute, was SARS. severe acute respiratory syndrome, 2003, caused by coronavirus. And why is that significant to Canada and specifically the city of Toronto? Because we were a hot spot. It was brought back from Hong Kong, and basically what was very fortunate about SARS, which was not true for COVID-19, was that it was such a severe infection that people had to go to the hospital right away, and really the outbreaks were mostly hospitals. But the hospitals were decimated. Many hospital workers died. Fortunately, it really did not spread to the general public very well. And so you didn't see the community spread you saw with COVID-19. But within a university, we still had to run exams. And it was somewhat problematic with some students who might have been coughing, et cetera. So that was really the first evidence of an issue with coronavirus. Going back up here, then in 2013, there was MERS, which was the Middle Eastern Respiratory Syndrome, which was also caused by a coronavirus. And it broke out in the Middle East, but did spread a bit, but again did not show the same infectious nature of the coronavirus. Because in reality, that virus was very good at infecting people. And for a virus to be really good at infecting people and spreading around the world, it has to have a lower death rate attached to it. Because if people die quickly, the pandemic weans out very, very fast. Okay. And then we have AIDS, which of course was in the 80s identified, but originally unsure what it was caused by, and really it's caused by the human immunodeficiency virus. And really, again, in the developed world is not really a problem anymore, in the sense that the multi-drug cocktail really does work. But where is it a problem and still epidemic?
Underdeveloped countries.
Sorry?
Underdeveloped countries.
Specifically Africa, because of the societal visions around HIV and who they think gets HIV. Do you know who the most common people who actually die of HIV in Africa are? Women. It's spread to women by men, and the women actually are dying at an enormous rate in Africa. And the biggest problem is that in many of the African countries, unfortunately, they would not allow the cocktail to be given because people didn't believe the concept of the HIV virus. We have established that HIV is totally controllable. Very few people die of HIV if they're diagnosed early. The cocktail literally takes that number of viruses and really turns it to a number below detectable levels. It can reemerge in individuals as they get older and perhaps become more immunocompromised. But it's a very excellent example of a success of antiviral treatment as to the success rate of survival of AIDS. But could never develop a proper vaccine to it. So they've tried to develop vaccines to AIDS, but they couldn't, the AIDS virus, but they couldn't. Lyme disease, which is Borrelia burgdorferi. You should be aware of Lyme disease if you live in southern Ontario. There's signs all over parks, don't go off the path. Because if you get infected with this particular bacteria and don't get treatment very quickly, it can lead to severe neurological conditions. And these are carried by, does anybody know? Ticks, a specific type of tick. And they've been traveling from the States towards Canada. They're now in southern Ontario. They haven't really found their way further north. So, for example, if you go a little bit further north into Muskoka, et cetera, you see less of it. But with global warming, they will be there, okay, because they can now overwinter. They never used to be able to overwinter. West Nile disease, which is actually a disease that, again, is carried by birds, and you can get by touching the birds. It disappears and reemerges at various times. So, for example, several years ago, Toronto had a major outbreak of West Nile disease. And if you have lived in this city for the longest time, you used to see all these big black crows that sat on the wires, and the entire population was decimated. And suddenly there were no more big black crows. Now they've started to reemerge, but this virus can take out birds, and you can get it by handling birds. And of course, that's also a problem with H5N1, which is a highly pathogenic bird flu, which is the last one, the highly pathogenic avian influenza that right now is creating havoc in dairy farms and in chicken farms. So this vision of the globe gives you an idea of types of infections that have emerged and reemerged. So this is from 2020, so you can see newly emerging viruses, and you can see the names of them in red. So in red are new emerging diseases. So obviously E. coli is not new, but the serotype of that E. coli is new. So you see an O and an H, don't panic. I'll explain what the O and the H means when we talk about the cell wall. It's characteristics of the cell wall. So you can also see reemerging or surging diseases and the location of these. And then you can see deliberately emerging, which is a little bit different in that and there's not really, there's only one example here, trace it back, and that's anthrax, that's bioterrorism. In other words, somebody intentionally created an infection. So we have constant surveillance for all of these emerging and re-emerging diseases. This is why the WHO, the CDC, Canada's PHAC is so important, because they watch what happens around the world, and they have to try and figure out what is going to be problematic before it really becomes terribly problematic. And one example of an emerging pathogen, which is a very interesting one, is Clostridium difficile. Its short form is CDI, and as a Clostridium, it's an anaerobic rod. And what happens with Clostridium difficile is that when you go into the hospital, many people who are in the hospital are undergoing immunotherapies or antibiotic treatment, and by definition, their normal bacterial flora gets killed off. That's not good for us, because that's bacteria protects us. And so what happens is you have your normal microbiota in your gut, etc. And as soon as you start to take large doses of antibiotics for long periods of time, you reduce the diversity of the microbiota in your gut. There is always Clostridium difficile spores in your gut, but they don't cause you any problem. Even if they germinate, they're in so small number they can't outcompete the normal population of bacteria in your gut, and they don't cause you any problem. But they do if all your really good bacteria have just been decimated by large amounts of antibiotics. And remember, the spores actually are not treatable with an antibiotic. They are dormant, but they then can germinate. And then they start to grow. And they start forming the Clostridium bacteria. And now this becomes an overgrowth, very hard to treat, very hard to get rid of. Many people actually do not survive this in a hospital. And so they have to try and control this in people. And one of the common ways it's controlled by is a fecal microbiota transplant. So they take fecal material from healthy people and they shoot it up their rear end, and they repopulate that person's gut flora. This is a common, by the way, it's not an uncommon treatment for a lot of people that have this condition, which is called dysbiosis, which means your gut has got out of its right combination. And as you do that, then in fact these bacteria can't outcompete, and the flora returns, and then the spores become not really very problematic anymore. But what it does is it causes what is called, because it's pseudo in that it's causing damage, it causes pseudomembranous colitis. So your gut just can't function at all. So this is another example of how treatment with antibiotics can lead to an emerging disease. And this disease has emerged a few years ago, let's say somewhere around 2010, 2012, and it's become very problematic in hospitals. We'll talk about flesh-eating disease caused by the group A strep, so don't worry about that. You'll hear that multiple times. I've already talked to you about the Mycobacterium tuberculosis, the multi-drug resistant tuberculosis. In order to clear this, when they give people drugs, they have to oftentimes put them up in residences and force them to take their drugs. Otherwise, the disease continues to spread. One of the things that is very problematic in the hospital, and you will be tested for it usually if you actually have a stay in a hospital. You'll be tested when you arrive, and you will be tested when you leave to see if you become a vancomycin resistant enterococcus carrier. And if you are, next time you end up in a hospital, you're going to be in isolation. So vancomycin-resistant enterococcus is just what it says. It's enterococcus, which is a common bacteria that's found in your gut that has become vancomycin-resistant, which is one of the most important last resort antibiotics we had. Okay. Similarly, MRSA, or methicillin-resistant Staphylococcus aureus, is the same scenario. It's Staphylococcus aureus, which used to be treatable with amoxicillin, a very simple penicillin derivative, and now is actually resistant all the way up to methicillin, which is was one of the newer forms of the beta-lactam antibiotics. And in the case, these first started to become a problem just before 2008, and in fact, you will be tested in the hospital. And one of the problems is, in 2011, they started to find these bacteria in bed bugs in hotels. So they had moved from the hospital setting to the community setting, which becomes even more problematic. So obviously, people who had been in those hotels were carriers of these bacteria. They got onto the linens, and the bed bugs bit the people. They ended up with the bacteria in their gut. And of course, bed bugs are very hard to get rid of. And that leads me to my last relationship to beta-lactamases. When we develop resistance to the beta-lactam antibiotics, which you'll learn much more about, they're the most common antibiotics we use for especially treating gram-positive, but also gram-negative infections. When bacteria acquire the gene for the enzyme, it becomes very hard to actually treat these infections. And there's been a variety of different modifications over time. I don't want to make it sound like it is just bacteria. It isn't. The 5th and 6th most common acquired infection in a hospital are fungal from Candida and from Aspergillus, and now the newest Candida auris, which is now on the watch list by most countries as being a potentially highly pathogenic form of Candida. Now, how many of you know your Latin? What do you think auris tells you where Candida was first found in somebody? Well, you're in the right destination. It's the head. It's the ear. Okay. So the first isolation of this was in the ear. And then you have diseases like measles that are reoccurring. Why are measles reoccurring? Why are measles, you have to go look at the Latin word for ear. Why is measles outbreaks reoccurring? It's a really simple reason. Yep. Yes, people not taking their vaccinations, or more importantly, people in their early 20s not getting their booster that has an added protection for you, because many of the childhood vaccines you had, the way in which they work, they start to lose their capacity. So therefore, you need a booster in your 20s to make sure you maintain to remind your body that you already have the information that you've had this infection before. And measles is not a simple childhood disease. It's actually a very serious childhood disease and a very serious disease in adults, and highly problematic in community centers like those in senior citizens. People will die quickly from measles under certain conditions, and it has a secondary condition, which is, does anybody know? It actually causes a severe inflammation of the brain. So it's not a minor little infection. It's actually a very severe infection, and it is the most, if not the most, one of the most contagious viruses there is. It is truly airborne, and literally, if somebody in this room has measles, a large proportion of you are going to, if you actually don't have, or either you haven't had measles or you didn't get your booster, you will get measles. Whole classrooms would be decimated because one person went to class with measles. So as much as COVID spread easily, measles is a whole other level of spreading. So the take home is we need to monitor these. We need to monitor hospital-acquired infections, which are also called nosocomial infections. Why? Because ultimately they end up in the community. They end up moving from hospitals to daycares and senior citizens' homes. And then they move, or long-term care facilities, or retirement homes. And then they move out further into the community, and they get taken home. And then they're spread, and now they're part of the community. Okay, so why do we need to get a flu vaccine every year? Or why should you get a flu vaccine every year? Why is flu one of these viruses that require you to get a flu vaccine every year? Because there are many different serotypes, and it changes every year. And what the companies try, the health, whether it's CDC or WHO, et cetera, they try to predict which strain is going to be the most commonly circulating. And the flu vaccine is not just to one of those. It's to whatever mix they think will be the most prominent flu vaccine that's circulating that year. Sometimes they miss, and so getting the flu vaccine doesn't fully protect you because the type that's running around isn't fully covered by that vaccine, but it still will protect you better than if you don't actually have the vaccine. All right. And we have problems right now with H1N5, which is actually infects birds. So we've had a problem since 2003, and it's been on and off, and then basically since 2015 has been problematic, but now we're in a major epidemic of bird flu. Fortunately, H1N5, again, has to do with the surface markers on a virus, which we'll talk about when I talk about viruses, is not able to go very easily from birds to humans, and has not really been able to go really human to human. But with a slight mutation, because it is any bird, pig, or human influenza has the ability to mutate when two different types of viruses come together, it could, in fact, mutate and become a major epidemic for humans. That's why we always watch the H1N5 grouping. Right now, it's just highly pathogenic, generally to dairy cows and bird flocks. And originally, it resides in wild bird flocks a lot, but sometimes it gets into bird farms as well, and then that's why, for example, eggs are going up in price, by the way, because the chicken farmers are losing so many chickens to the bird flu. H1N1 actually which was a major flu epidemic. H1N1 was a mix of a bird influenza virus, a swine virus, and a human virus. So it jumped from humans and then could go humans to humans in 2009. So that was the last major flu pandemic in 2009. It was highly problematic, and it was highly problematic to the groups of people that normally do not get the flu. Usually flu is highly problematic for the elderly, obviously the immunocompromised, and the young. But in this case, the majority of people that actually had severe flu were actually healthy people in their late teens into their 20s. And the reason is because generally if you were older than that, several pandemics have been of the H1N1 type, including and it's called the Spanish flu for no good reason. It's a flu that actually developed as World War I was ending in Europe. And of course, as the war ended, what happened to all the soldiers, etc.? They came home and they brought the flu to their various locations. And it had a substantially high death rate to it. This particular outbreak in 2009 was very similar to the Spanish flu, as well as the one that occurred in 1968. So people who had had those flus and or been vaccinated were better protected. So the elderly happened to have gone through at least the late 60s pandemic, so they had some protection against it. But the younger people had no protection against this particular flu strain. And we've had problems with a variety of different avian flus. If you're interested in this, we'll talk a bit more about it, but watching avian flus is very important. But what we learned during the pandemic was the following: watching bird flus at the expense of not watching problems that can come out of areas where we have animals mixing with people where they usually don't mix, we can miss potential epidemics. And really, the coronaviruses should be on a list. By the way, you all probably have had a coronavirus and not COVID-19. Generally, coronaviruses give you the common cold. So these are specific types of coronaviruses. So they're commonplace all over coronavirus is common, but the particular ones that have caused the pandemics are not common. And really, they should be watching coronaviruses because they've established that they have a strong ability to cause major pandemics as well. All right. So we're just going to remind you and put everything in context for us to move to the next topic after the break. So if we really look at the major discoveries that we've talked about to set ourselves up for the rest of the term, we've talked about this and the role. We're not going to talk about, as we move past general microbiology, we moved into an era of molecular biology and general microbiology that allowed you to determine the genetic code, etc., discovery of archaebacteria. And now we really have moved past even this molecular microbiology, genomics, and proteomics into a lot more big data driven experiments where we try to identify characteristics of microbiomes, characteristics of unknown diseases, etc. So with that, your in-class assignment, which really is on Quercus, it should be live now, is to summarize for a first-year biology course the important discoveries, role of the discoveries from the mid-1600s to the late 1800s. I've listed the names. Do this in it's not a long assignment. Just use 4 to 5 sentences so that a first-year student will understand the relevance. And by doing that, you understand the most important discoveries of that era that still are important today to the way in which we actually carry out science. Okay, so if I remember what I did, I believe it's live until Monday at 10 p.m., but you can double check on the assignment. Okay? All right, so we're going to take a break and we're going to switch topics now into evolution of microorganisms and diversity, which is going to set us up for understanding bacteria characteristics. So I have 10:09, so come back at about 10:19. So we're going to move into the next topic, which we will cover today and into next week. And I'm using the evolution of microorganisms to really introduce you to the diversity of characteristics of prokaryotes and really just step into the role of prokaryotes in the evolution of the origins of eukaryotic cells, which is the endosymbiont hypothesis. So we will, during the course of this time, ask a question: where did the first cells come from? And address the various characteristics of bacteria. And I'll put it in context to the endosymbiont hypothesis. So, excuse me, if we look at the current hypotheses of the origin of cells, really, what we first learned is that the experiments of Spalzani, Pasteur, and others demonstrated that under the conditions that exist on our planet today, living cells do not arise from non-living matter. But what we have to turn our attention to is what the Earth looked like when the first cells evolved. And the Earth did not look like it does now, right? So under what conditions did the earliest cell types arise? What were the characteristics of these early cell types? And how did the diversity of cell types that exist today evolve? So the hypothesis ultimately is very straightforward, which is that all cells have descended from a common ancestor that was the universal ancestor. So as I go through this, I will tell you right now that, yes, I took microbiology a long time ago. This was not the hypotheses that I learned when I took microbiology in the third year of my undergraduate education, because we did not know that RNA had ribozyme activity. And so the hypothesis was turned on its head when we realized that RNA-based cells could carry out enzymatic activity, because some RNAs have the ability to be enzymes. That was a discovery into the 80s. Okay, so things do change based on discoveries. So the basic principles are the same, but some of them are different. So the present conditions of our planet, they don't support development of living matter from non-living matter. However, the conditions that existed at the time of the origin of our planet, around 4.6 billion years ago, to approximately 4 to 3.8 billion years ago, when the first cell-like systems are thought to have evolved, were very different. So what happened in the first 6 million to a billion years before the first cell types arose? Well, based on molecular biology, geology, astronomy, they all suggest that prokaryotic-like organisms basically have been on Earth for approximately 3.7 to 3.8 billion years ago, and that's based on fossil evidence in the stromatolites, which are sedimentary rocks that are formed by cyanobacteria, found very often in the oceans. This is a stromatolite, by the way. So by dating this, they can start to realize when the first organisms likely evolved. So if we remind ourselves, and this is a microbiology course, so I don't want to go into vast detail about various aspects, but we have the origin of Earth down here, and basically the oldest prokaryotic fossils in around 3.5 or 3.6 million years ago. And we have, just prior to that, the oldest chemical evidence of life. So what happened during this period of time to allow for these prokaryotic cells to evolve that then allowed, the first eukaryotes to evolve, which also coincided with the first oxygen-producing cyanobacteria, and then ultimately multicellular eukaryotic cells. So for almost 2 billion years, prokaryotes were the only living inhabitants on this planet, and hate to break it to you, but they'll be still here after we're gone too, because they're very adaptive and we aren't particularly adaptive. And during this early time period, but also true today, prokaryotes extensively shaped the conditions of the planet and its atmosphere. So the diversity of the cells we see today, when you look in your microscope and do the biochemistry, really evolved from the prokaryotes. So we can put this, in connection with when oxygen started to become available and the various eons. And what we really see is that the first evolution of the first cellular life, which was LUCA, really occurred at a period of time when the Earth was not, there was no free oxygen, right? And so basically, the conditions at this time on Earth were very different than they are today. So we were not in an oxygenated world. We were in a very different scenario. And as we moved out of this period where we developed the first cells, which were anaerobic, so this is where you see that this area is anoxic, so these bacteria were predominantly anaerobes. With the evolution of the cyanobacteria, which are really, they are bacteria that people often labeled blue-green algae, but they are not algae because, of course, algae are eukaryotes. They live in lakes, streams, ponds, etc. And they evolved the ability to carry out classic photosynthesis as you know it. A photosynthesis that uses CO2 and produces oxygen. And that led to the oxygenation of the world. And then the other cells, various cell types could evolve because we created conditions as we move through increasing oxygen levels to reaching a point where we could support aerobic respiration. So what are some of the theories? Well, for the most part, the first cells would have been some kind of self-duplicating system. And then they would eventually become living cell-like systems. And these would have perceived to have evolved by what we would define in terminology as being prebiotic or abiogenic or abiotic process. So in other words, they were chemical reactions. They were not biological reactions, okay? So prebiotic means before life, abiogenic means not generated by a biological system, abiotic means not involving biological systems. So basically, chemical reactions occurred to make the building blocks of the first self-duplicating systems. Remember, anything that we make in a cell can be made on a lab bench. All we have is enzymes that speed up reactions. There's no reaction that a living cell can do that can't be done chemically, but often under very harsh conditions, okay? So keep that in mind. I think we tend to think that, enzymes are they are magical, but all they do is act as catalysts to increase the speed of reactions, okay? So when we think about the evolution of the first universal ancestor, the centionote, which would have been the first DNA-based cell from all liver cells, is believed to have evolved from RNA. And that change in that, if you go back and read old textbooks from the 70s, this would not say this. Even into the late 70s, it would not say this. As the discovery of ribozymes were being presented at scientific meetings, that's when they had to start changing the theories. And that was really evidence of the enzymatic activity and the ability of self-splicing RNA that led to this discovery. Okay. So basically, if you have chemical reactions occurring in the absence of users, yes, it could take hundreds of years to produce a single usable material, but it's under conditions where there were no users present. So over time, you would have accumulated abiogenic products in the absence of users and under anoxic or no free oxygen conditions. At some point in the evolution of the primitive Earth, organic and inorganic substances are thought to have become sufficiently concentrated to start allowing frequent interactions. And these reactions would have adhered to the clay or other particles like pyrite, which could help catalyze those reactions. And that, by the way, this would have also been occurring in hot sulfur-rich environments, right? Because that's what the environment would have been. So these microenvironments that would allow for catalyzing reactions are proposed to have given rise to the first RNA-based system. So in the first billion years or so, we have to think about this. And first of all, there's an order to this pathway, but it's not like this pathway was the only way it took. There would have been multiple ways in which things evolved, and really what we can identify as being the likely predecessors were those that survived. Because things were happening all over the earth. They weren't just happening in one little place, okay? So don't think that everything went boom, 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 just as I'm describing it. What I'm describing is that which would have survived and been successful in actually carrying out function. So the important thing we have to realize is this is not spontaneous. We would have had evolution of various types of self-duplicating systems within the first 200 million years after the formation of the planet. And these early cell-like systems, or the prokaryotes, are thought to have evolved around, as I said, about 4.4 billion years ago, as self-duplicating RNA molecules becoming enclosed in a lipophosphate membrane. That was enough to create a so-called cell that could function. A membrane, RNA, that could duplicate itself with the chemical material that was surrounding it and could also carry out enzymatic reactions. Then, somewhere around just before 3.8 billion years ago, the first DNA-based systems would have evolved, and we have evidence, fossil evidence of these in sedimentary rock. The sophisticated, if you want to call them, prokaryotes, the photosynthetic bacteria, have been observed in rocks that are approximately 3.5 billion years ago, and fossils resembling the present-day cyanobacters are thought to have evolved by about 3.1 billion years ago. So this is kind of just a diagram of one person's vision of this. So you have chemical reactions, nutrients in a hot hydrothermal water. You get prebiotic chemistry that's making a variety of potential building blocks. RNA evolves. It can carry out both division of nuclear material, could carry out enzymes. So you could have cell division. And then as that evolved over time, eventually DNA evolved. And one of the major reasons going to be is that RNA was very unstable. And RNA is not a good genome for stability. And ultimately, you end up with the first DNA-based cell that then leads to the early bacteria and the early archaebacteria, which they will continue to debate whether they came off at the same time from the centrozoa or the archaebacteria came off just a little bit after the eubacteria evolved. And of course, that was another discovery. We didn't know archaebacteria existed, so the original models didn't have archaebacteria in the models at all. So we think about what the conditions were, and they were under reducing and anoxic conditions with little or no free oxygen. These conditions no longer exist. We're in an oxygenated world. So reducing conditions have not really existed since the evolution of the cyanobacteria around 3.1 billion years ago. But the key point of all this is in the absence of users in that previous slide is you could make amino acids, you could make DNA or RNA, et cetera, because they were just chemical reactions. You could make carbohydrates, et cetera. So in the absence of users, there were no or very few systems existing that could take up, metabolize, and otherwise disassemble these substances at a rate faster than they could be made by abiogenic synthesis. But obviously, as you evolve cell types, abiogenic synthesis is not going to be able to keep up with the need for building blocks for cells. Okay? So if we kind of put this in a table, we start out with the type of evolution as being the formation of this Earth about 4.6 billion years ago in the absence of free oxygen, so anaerobic and reducing. We had chemical evolution, the RNA-based systems with creating building blocks with very few users until about 3.8 billion years ago. And then we had biochemical evolution, where we saw a split of anaerobic bacteria to give the two cell lineages at around 3.8 billion years ago. starting with anaerobic photosynthetic eubacteria. So yes, anaerobic bacteria photosynthesize. So photosynthesis is just not as you've learned it, using CO2 to produce oxygen. Okay, bacteria do it slightly differently, and you'll see that basically the process is the same, but the starting or the building block in the end product is different. Okay, and then you moved into scenarios where the cyanobacteria started to be able to use oxygen started to become a potential product based on a change in the way in which they carried out photosynthesis. I'll show you that diagrammatically in a moment. And then you got the evolution of the non-photosynthetic aerobic autotroph eubacteria, which are called the chemolithotrophs, and then heterotrophs actually also started to evolve. So we needed that free oxygen to carry that out. So if we look at the first sen ancestor, it would have been prokaryote. So what does prokaryotic mean? Simple cell architecture, no nuclear membrane. The first cells would have been anaerobic and heterotrophic. Okay, they weren't photosynthetic, they were anaerobic and heterotrophic. And then the cyanobacteria evolved to create oxygen, and then the aerobic heterotrophs would have evolved. Heterotrophic means requiring an organic carbon source for growth, and anaerobic means they do not use oxygen in metabolism, and more importantly, anaerobes are generally killed by the presence of oxygen. Many modern-day bacteria, of course, fall under this category of being prokaryote, heterotrophic, and anaerobic. They're primarily the eubacteria. Almost all bacteria are prokaryotic and heterotrophic, but they have evolved to be aerobic rather than anaerobic. So what does it mean to say eubacteria? They can be photosynthetic or non-photosynthetic. They can be heterotrophic or autotrophic. They can be anaerobic or aerobic. What we have to remember is that the non-photosynthetic, heterotrophic, anaerobic eubacteria are thought to have evolved directly from the sen ancestor. So the first bacteria would have been non-photosynthetic, heterotrophic, and anaerobic. Then the photosynthetic anaerobic bacteria would have evolved. They had to evolve for the classic the classic organisms that can carry out the photosynthesis that you constantly are reminded of, including in the trivia night last night, if any of you were there. All right, so archaebacteria, they split off very early from the non-photosynthetic heterotrophic anaerobic eubacteria. However, some of the diagrams will show an immediate split from the sen ancestor, and that still remains a debate as to when exactly the archaebacteria split. Was it an immediate split, or did the archaebacteria come out of the first eubacteria that evolved? The direction is to say it was a direct split, but that still remains a mystery. The cyanobacteria, which often in any book or often says blue-green algae, it's an incorrect term for them. They are not algae. They are bacteria. They are eubacteria. And they're thought to have evolved from the photosynthetic eubacteria that were anaerobic, but ultimately developed the ability to carry out photosynthesis, taking CO2 and converting it to oxygen. So the cyanobacteria kind of sit where the evolution took place. So many cyanobacteria can carry out both anaerobic photosynthesis and aerobic photosynthesis as a result. But we have common shapes, those shapes that would have evolved early on. We already discussed that a bit last week. We have the cocci or the coccus. We have the rods. We have Spirillum, which are spiral bacteria. We can have spirochetes, which have a number of curves. And we can get some variations of stalks and hyphae. And then there are some bacteria that can grow as a rod, but then can form very long filaments. So they actually don't separate their cells. We won't actually see this type of bacteria or see this kind of growth in any of the bacteria you look at. A filament is different than rods that are found in chains. So Bacillus often is found in chains. You would have seen that with Bacillus megaterium if you got your negative stain to work. But it's not a filament. It's a series of rods that are in chains. So the key stains in terms of being able to identify different types of bacteria are your acid-fast stain and your Gram stain. Now, what you see in your Gram stain here, colors a little, so the darker color is really the purple color. These are cocci. This actually would be Staphylococcus, and you can see the tiny red rods, which are actually E. coli. Now, those of you who were trying to do your hanging drop slide and you kept seeing these long filaments, which was in the media, that was not the E. coli, because E. coli is a tiny little coccobacillus. It looks like a little bullet. Many of you think it's cocci because you perceive in your brain that by definition a rod is always bigger than something that's round. That is an incorrect assumption. So E. coli is a short little rod that has defined sides. There are cocci that are much bigger than E. coli. Okay. Here you see an acid-fast stain, and what you have is two different types of bacteria. This is Mycobacterium, which is a very long, thin rod that is actually acid-fast, so it's maintaining the first or the primary stain, which is carbofusion, which you'll read about as a demo and a read-only and a demo slide next week. And in the background, you see the non-acid-fast bacteria that take on the secondary dye, which is usually methylene blue. Now, don't get hung up on the names of the stains. Safranin, methylene blue, crystal violet, carbofusion, they're all basic dyes, which means they will stain the negatively charged cell walls of gram-positive and gram-negatives. The reason you pick particular combinations of stains is for visual purposes. So if you want to differentiate gram-negatives from gram-positives, you really don't want to use a green and a blue stain because it becomes very hard to differentiate. So you use crystal violet, which gives a nice purple or blue color, depending on your color definition, and safranin that gives a red color. So almost all the stains, except for the negative stain, which stains the background, and the capsule stain, where you'll use a negative stain, they all stain bacteria. They're all basic dyes. The color is just being chosen to make it easy to differentiate. Okay? And all bacteria, whether they're gram-positive or gram-negative, have a negative charge. And we'll talk about why, but it has to do with the components in their cell wall. But there are things you will see in your staining in terms of your staining, which are not bacteria. So if you see a common precipitate that has a pattern, like the one on, these are not bacteria. It's a stain that started to precipitate. It often occurs in an acid-fast stain. What often can occur in safranin and crystal violet are crystals, because the stain starts to precipitate and it forms crystals. There are no crystal-shaped bacteria. One year a student put up my hand and said, Dr. Brown, I have a really big bacteria that looks like a star. And I said, oh, let me see. Oh yeah, those are very nice crystals. Okay, so look beyond what's there. Bacteria are small. They are not big crystals. If you have a stain that has a lot of crystal in it, please tell your TA and they'll get you a new bottle of stain to make things easier, because it's really fascinating the kinds of shapes crystals can be, but we're not studying crystals in microbiology. Okay, so keep that in mind. There will be artifacts in your stains. So you did a negative stain. This is a negative stain showing a capsule that's counterstained to make the capsule more visible. So here you can see this is the rod. Here's your big capsule, and the background is stained. In this case, there was a staining done to stain the bacteria as well. And this is a slightly different stain. This is the capsule stain you'll carry out in the lab. And again, the background is stained. You then counterstain to stain the cells. And generally, the capsule remains unstained. The capsule is usually wider than the cell. So sometimes you'll see a hint of a capsule. It's not a real capsule, it's an artifact. Capsules are sticky polysaccharide that are really thick, that allow bacteria to stick to surfaces. So it really needs to be a very thick capsule for it to really truly be a capsule. And if you do too much rinsing of water, because capsules are made out of polysaccharides, which means they are water soluble, you will wash the capsule off. That's why in capsule stains, you don't really use much water. And the reason you don't get to keep your capsule stain is because you don't actually heat fix your slide, because if you do, you create all sorts of artificial capsules. So that's why you have to show your capsule to your TA, and then he or she keeps a chart that says that they saw your capsule. We'll talk about endospores in much more detail, but you'll do your endospore next week. So here is Bacillus cereus. And if you look really carefully, the vegetative cell is pink and the endospore is green. And so as the endospore develops, it forms within the cell. And then once it's mature, the vegetative cell dies and all you see is endospores. So you can see some of the endospores here that have been released from the cell. You can see with Bacillus cereus, you can see the position of spores is genetically determined. So if you look carefully in Bacillus cereus, the spores are in the center. If you look at Clostridium tetani, here is a spore that's been released. Here is let's find a better one. A cell that, here is a cell that has a little bit of a tail on it, as does here. Clostridium tetani, the spores form at the end of the cell. So there is a genetic component to where endospores form. They do not move around. They are only found in a, so you can use the endospore to help identify the bacteria. And in the case of Clostridium tetani, the spore is bigger than the cell, so it looks like a drumstick, or a popsicle as a result. All right. So those are, you're not going to do, you'll do an endospore stain in week 3. You're not going to work with Clostridium tetani because A, it's a pathogen, so we'd have to be a level 2 containment, and it's also an anaerobe, so unfortunately, the conditions to grow them is very difficult. To remind you of the common features of modern prokaryotes versus modern eukaryotes, you notice there is no nuclear membrane. You simply have an area called a nucleoid, which is the free DNA. But it is in close proximity to all the ribosomes. And you will notice that the bacteria are full of ribosomes. They are protein-generating machines. And because their DNA is not separated by a nuclear membrane, as the RNA is made, the ribosomes can immediately engage and start making protein. So the speed at which prokaryotic cells can make protein is much faster because you don't have to make mature mRNA that gets transported to the cytoplasm, which then leads to translation. There are no organelles, but you do find a few storage granules, which I'll demonstrate in a minute to you. And you will find in bacteria some autonomous circular DNA, plasmids. Most bacteria have double-stranded plasmids in them, which carry out ancillary roles in the bacterial cell. And we'll talk about that in much more detail. Some of them only replicate when the chromosome does. Others replicate independently. I don't want to go through this in any detail, but this is your Gram stain, very important steps. So in this course, you're not going to be asked for timing. You're not going to be asked for, but you will be asked, well, what would happen if you didn't add, for example, iodine to your Gram stain? So you have a stain. And what you do is you flood with the primary dye. I told you a primary dye stains all bacteria. So if you have a mix of gram-negative and gram-positive, they'll all get stained by the crystal violet. You established that last week, by the way, if you did all your staining with crystal violet. Okay. Then you will add iodine, and both types of cells will still be stained. Iodine is a mordant. It'll form a complex with crystal violet. And that mordant will help keep the crystal violet in the cell. And it will be selectively keeping it in gram-positives because it'll more easily wash out of gram-negatives. Then you add the decolorizer. And the decolorizer, for that short time, removes the stain from the gram-negative, but the stain in the gram-positive is harder to remove because of the nature of the cell wall. And so now if you look at the slide, you would see no gram-negative cells stained, and you'd only see gram-positive stained. So if you forgot the iodine, and went and tried to decolorize, then in fact you would see no cells because the crystal violet would wash very easily out of the gram-positive as well. And then you counterstain with another basic dye, which is safranin, which then stains the gram-negative, and these are just some examples of gram stains. So you've already done this last week. You prepare a smear, you dry in air, we heat fix on a, we dry your slide on a slide dryer, and then you flood with the stain, you carry out your stain, whatever it is, you look in the microscope, and you're going to use the 100 times objective, which has, with the eyepiece, another 10%, which is a 1,000-fold magnification. And you need the oil, because the oil allows the light to go directly through the slide and into the objective. So otherwise, the light scatters. So the oil does not increase magnification. What does it increase? Resolution. Yes. Okay. So some other common features of modern-day prokaryotes. The nitrifying bacteria, which we talked briefly about last week, will have all these membranes from which they can carry out nitrification. So these are thylakoids. Photosynthetic bacteria will have a lot of membranes by which they will carry out photosynthesis. Here's just an EM picture showing you the cell wall, the cell membrane, and the nucleoid, and then the large amounts of ribosomes. So there are some variations. You can see large amounts of membrane in the photosynthetic and the nitrifying bacteria, depending on the conditions you find them to grow. You don't really find very many subcellular structures, but the ribosomes will be about 25% of the dry weight, which is very high. And again, you'll have a chromosome and likely plasmid DNA. But some will have what are called inclusion bodies, and those inclusion bodies will be based on growing in particular media. So what bacteria will do is if they find themselves growing in a high concentration of phosphate or sulfur, they will store that for future use, so they'll create granules that they can use for nutrition purposes. So they can store fat, polyhydroxybutyrate, sulfur, and polyphosphate are the three most common that they store, and some of this can be diagnostic. So here you see some staining. Here are some polyphosphate granules. Here are sulfur granules in one type of bacteria, and here are some fat droplets in some bacteria. In each case, they would have had to be growing in high sulfur or high phosphate or high fat to do that storage. So you don't always see them. All right. All right, DNA replication, transcription, and translation all take place in the same cellular compartment, which I said earlier, in the cytoplasm, which allows the bacteria to do things quickly. The genome, the DNA of most genes are haploid. So bacteria, by definition, are haploid. They are not diploid. However, they can have some additional copies of the same genes on plasmids within the cell, which means for some genes they may be diploid, but they're being carried on two different DNA structures. Most genes lack introns. So bacteria don't have introns, which means they don't undergo RNA splicing generally, which also speeds things up. Introns are very rare. Occasionally they occur. They're self-splicing. So if you remember your B11, you have self-splicing introns, which can splice themselves using ribozyme activity. And then in higher eukaryotes, you have a spliceosome that has ribozymes and RNAs and proteins that help actually splice the introns out, that occur where in a eukaryotic cell? Where do introns get removed? Sorry? In the nucleus, right? What leaves the nucleus is a mature messenger RNA to which the cap in the poly A tail has been added. Eubacteria and cyanobacteria, but this is an important point and comes to the evolution of eukaryotic cells, but not the archaebacteria, contain only one major DNA-dependent RNA polymerase. And it's responsible for transcribing mRNA, tRNA, and rRNA. So they don't have different polymerases for transcribing different RNAs. But the archaebacteria do. all, but remember, also contain all organisms also have a minor DNA-dependent RNA polymerase, which is the primase, which is involved in making the Okazaki fragments during DNA replication. All right. What about the archaebacteria? Well, archaebacteria are prokaryotes. Therefore, they have all the previous features that I identified, except where I gave you some exceptions. But they have several DNA-dependent RNA polymerases in addition to the primase. But the interesting thing is a lot of them cross multiple functions. So they're not like doing very specific functions in the archaebacteria, but they have multiple RNA polymerases, which is one of the evidences that the archaebacteria is the predecessor to the nucleus. So their structure is somewhat similar to the eukaryotic RNA polymerases, which we'll talk about in a few lectures. The other interesting thing about archaebacteria is that, in fact, many of them have multiple origins of replication. So prokaryotes have a circular DNA, a double-stranded DNA with one origin of replication, the eubacteria. Eukaryotes generally have linear DNA that's double-stranded with multiple origins of replication. The archaebacteria have, just have to make sure if it is recording. This is okay. The archaebacteria actually have, some of them, not all of them, have multiple origins of replication. This again is another hint that the archaebacteria are likely the predecessors to the eukaryotic nucleus. Morphologically and physiologically, the archaebacteria have morphology similar to the eubacteria. In other words, their shapes are similar, but their structure of their cell walls, the structure of their membranes, their biochemistry are very different. And I'll come back to that at various times during the course. The reality is that when we compare an archaebacteria to a eubacteria and to a eukaryote, what we discover is that archaebacteria are as different from eubacteria as they are from eukaryotes. So for example, the genome of the methionine-producing archaebacterium Methococcus genancea, which has been completely sequenced, shows 44% of the genes are encoded proteins that resemble that of a eubacteria, but 56 of them are totally unique to the archaebacteria and never seen before. So in other words, yes, they do share about 40% similar genes to the eubacteria, but over half of it shows no similarity to the eubacteria, and also do not show similarity to the eukaryotes. So again, sorry. So what this suggests is that the way in which they grow, or where they grow, has obviously been very important in how they've evolved the genes that are, and the gene products that will allow them to grow in those environments, because eukaryotes don't grow there and the eubacteria don't grow there. So most archaebacteria, as we've already discussed, have very specialized habitats. Avoid the text table information that's left over, I apologize. And they grow in very extreme environments like acidic, hot, hypersaline, anaerobic. But there are some eubacteria that can grow in what would be defined as extreme environments. There are many bacteria that grow happily at 65 to 70 degrees Celsius. Now that would be a low end for an archaebacteria, but it's still a very high temperature. And in fact, we've relied on them for the development of the thermostable DNA polymerases we use in molecular biology. So the archaebacteria are truly extremophiles, and there's a subpopulation of the eubacteria that can grow in some what we would define extreme conditions. So what do we mean by extreme? Well, thermoacidophiles can grow at a pH of 1 to 2 and above 100 degrees Celsius. So has anybody ever dropped HCl on their hands? I hope not. Because what does it do to your skin? It burns it, and that's the pH at which the HCl is at. 100 degrees Celsius is boiling water. Okay, so that's a very extreme condition. The hypothermophiles can grow in the walls of deep sea hydrothermal vents, which are rich in sulfur, and they can reach up to 300 degrees Celsius. Some of these archaeobacteria can grow in 4 molar salt. So while we thought that nothing could grow in the Dead Sea, they're there, or the Great Salt Lake. And they also can grow on salted foods. They also can be alkaphiles, which means they can grow in very alkaline conditions. So in lakes that reach pH of 8 to 10, again, that's a very high pH. And they can grow in very cold conditions like an Antarctic or the freezer. So by the way, for suffix, phile in the world of biology means lover. Therefore, in microbiology, it means they grow best. It's their fastest duplication. If you see troph, it means it's capable of growing in those conditions, but it's not the ideal conditions for it. Just to finish really quickly, there are some modern day characteristics of the cyanobacteria, and this is where we'll pick up next week. So that the cyanobacteria sit in that border from going from the anaerobic photosynthetic to the aerobic photosynthetic. So many of them, when they have sufficient light available, they're photosynthetic and autotrophic. Some of them, when insufficient light is available, they're heterotrophic. When oxygen is present, they carry out photosynthesis similar to higher plants and algae. When oxygen is absent, this is what's really interesting, is a small population of cyanobacteria can carry out types of anaerobic photosynthesis seen in the photosynthetic bacteria. And many of them fix atmospheric nitrogen gas to ammonia, which provides important nitrogen to biological systems. So therefore, they do have extensive membrane systems called thylakoids, where the pigments and electron transport chains are also that photosynthesis is located. And an important point, cyanobacteria, some of them produce a range of toxins, many of which are very harmful. So they, just like algae, are responsible for severe algal blooms in lakes and can produce severe toxins that can cause you severe irritations. And if dogs or other animals are playing in that water, they can kill those animals. So if you see an algal bloom, don't go swimming there because it may be producing toxins. All right, I'll pick up where we left off today next week. See you next week. Don't forget to prepare for the lab.